Hello, and in this particular video, we will learn how pure lines are generated, how they are created. In our previous video, we learned about what pure lines are. Pure lines refer to a population that breeds true for a trait or for many other traits, right? But now, how are these pure lines generated? But first thing first, why generate pure lines? Now, pure lines are commercially important for plant breeder or animal breeders. For example, if a person is able to develop a pure line for a particular plant, every time it self-pollinates or cross-pollinates within its own type of members, it will always produce the same characteristic for a particular flower, let us say flower size or flower color, etc. Similarly, pure lines are also important for plant breeders. For example, a person has a particular breed of a cow which gives a very high quantity of milk, and if he has the same pure line, which means even the male counterpart has the same kind of genotype inside it, then the reproduction will give result to cows. Whenever the female is born, the cows with the same characteristic, they will always produce higher quantities of milk. So pure lines are important commercially for plant breeders and animal breeders, and also for geneticists for genetic experiments. Now, how are pure lines generated? Let us try to understand it by an example. So, uh, I'm taking a particular plant here which has the following phenotypic characters. So in a particular population, a particular plant produces red flowers and yellow flowers. And let us assume that the genotype here is capital R small r. Capital R is the dominating allelic partner, and whenever it is present, it will give rise to red flowers. However, the allelic partner small r, when present with another recessive partner, will give rise to yellow flowers. Now, how can, let's say, a plant breeder generate a pure line which will only yield red flowers? Or let us assume only breed yellow flowers. For the sake of simplicity, let us just consider that the plant breeder intends to only get red flowers because they are like really, really commercially feasible or useful for the plant breeder. Now, in order to generate pure lines, two uh, things are done. And the first is two actions are taken. First, selfing is done. The plants are only self-pollinated, and they will not produce a pure line only after the first generation or in the first generation. So the self-fertilization must be conducted generation after generation after generation. How long? Until the desired phenotype is conserved. And we will see what I mean by whatever I've said just now by an example, by basically building up a Punnett square and a few other graphical illustrations. Also, another thing that is done to generate pure lines is the process of selection and deselection. Plants that do not exhibit the trait of interest are deselected, are chucked out, whereas the plants that have the desired trait are kept and further self-pollinated until the development of the pure line. Now, assuming that, let us just take this example. Now, if this plant is selfed, what do we mean by selfing here? It means that the pollens from the same plant will be used to fertilize the eggs of the same plant. So let us assume that this here is the genotype for the male partner, and this is the genotype present in the female partner, basically in the androecium and in the gynoecium. So let me just put a little symbol on the top which represents male and female. So I'm considering this as the male and here is the female, right? It doesn't matter though, but let's just write it here. So what will be the outcome? The outcome during the process of gametogenesis is half of the gametes will contain the genotype capital R, and half of the gametes, which is 50% of them, the male gametes or the gamete present in the pollen grains, will have the genotype small r. Likewise, in the female part, which is basically the ovary or the inside the ovules, we will have two types of them. One will have half of them will have the genotype capital R, and half will have the genotype small r. Now, these are the combinations, and we wish to study what happens when they are self-fertilized, when self-pollination takes place. So in order to understand or figure out the genotype and the phenotype of the first generation, let me write G1 for first generation, P1 for the first generation phenotype of the first generation. Let us see what the outcome is, and for that we will use a Punnett square. Now the males are written onto the top and the females are written to the side. This is the basic convention, but in this particular case it doesn't matter because both the outcomes are the same. Capital R, small r in equal ratios. So let's just write the outcome. So here goes the male gametes to the top right here, and let's write it down here. So this is capital R and small r, and then the female gametes onto the side, capital R, small r. And the combination here in the first square is capital R, second square is capital R, small r, and the third square is capital R, small r, and in the fourth square it is small r. Now these are the four possible outcomes. So the breeder gets the seeds, and then what does he get? Let us just write the genotypic ratio. Now, how many genotypes do we see here? Let me use a yellow marker to just circle the types of genotype. So we get this particular genotype, capital R, small r. Is this repeated somewhere else? No. So we have one genotype called capital R, capital R. The second type of genotype is capital R, small r, and we got two of those. One is here, capital R, small r. And the third genotype is small r, small r. So from the parents with the genotype, from the parents that had the genotype capital R, small r, we get three further genotypes. One is the same as that of parent, capital R, small r. The other is capital R, and third is small r. And let's just write the three genotypes here. R, capital R, small r, and small r. Now, what are the three different phenotypes, or two, or whatever number of phenotypes? What are the phenotypes that we get? So if you look at these three here, these three genotypes will have the same phenotype. Why? They will all produce red flowers. Why? Because all of them have at least one dominating allelic partner. But if you look at this fourth one here, right, the fourth one is homozygous for the recessive allelic trait. So we will have, out of four possible combinations, we will have one combination that will produce a yellow flower. So we've got two phenotypes here. We get red and we get yellow flower producing plants. So how many red producing plants do we have? We got like 3. And how many yellow producing plants do we have? 1 out of 3. Now, I hope you remember what are we doing. So we are conducting self-pollination, and now we've got an outcome, and now we'll move to this particular process, which is called select and deselect. So do we need these particular plants? Well, the plant breeder is not interested in generating yellow plants, right? So he discards all of these and continues with his experiment with these 3 particular plant types. Now, let us so we have already seen what happens when we self capital R and small r. So out of these 3 plants, you see here, out of these three plants that will produce the red flowers, we have got 2 different genotypes. There is one genotype which is basically homozygous for red flowers. Have a look here. So one of the 3 is homozygous for the red flower, and 2 of them are heterozygous for the red flower. So we know the outcome. What will be the outcome of a heterozygous selfing? If we take these 2 plants and we self-pollinate them in the second generation, or rather, sorry, in the first generation plants, if they are self-pollinated, we know the outcome. Again, the outcome will be the same as here. We'll get 3 red flowers and 1 yellow flower producing plant. But let us just assume the plant breeder again. Now he has no choice. What he does is he takes these 3. He will right away discard the yellow, but he'll take these 3 genotypes, and let me just write them separately, slightly separately. Even though they are the same, let me just write it like RR and RR. And what he will do, now he has got these 3 types of plants, and he will self-pollinate these, he will self-pollinate these, and he'll self-pollinate these. Right, and probably he'll do it on 3 different fields, right? So this is one field, this is the second field, and this is the third field where he'll be doing this. And in this particular field, what will be the outcome? In this particular field, what will be the outcome? Here, in this particular field, the outcome will be, again, we will have 3 reds and 1 yellow. Here again, 3 reds and 1 yellow. And why do I say this? Because the genotype here is the same genotype as that of the parent, and we've already done the Punnett square outcome for this. And so we see that in these two particular fields we'll again have three reds and one yellow, three red and one yellow. But what will be the outcome if we self-fertilize this particular genotype capital R and capital R? Even though it's pretty easy, let us still build up a Punnett square and solve it for ourselves. Now, if this particular genotype capital R has to undergo self-pollination, one of the partners will act, the same plant will act as a male, the same plant will act as a female. So the male part will produce gametes with the genotype capital R, capital R. Half of the gametes will have this particular dominating allele and the other half will have the other allelic partner of the pair. Well, it doesn't matter, both are the same. In the females, again, it will be the same. Each egg will have a capital R here and a capital R here. So 50% of the eggs will have one of the allelic partners and 50% will have the other allelic partner. Now, what is the end result if the selfing or self-pollination takes place here? So let's write. So here we have the male partners. Let's write their genetic or genotype on the top. So capital R, capital R, capital R, capital R. And now this is the second generation for this particular allele, for this particular combination or outcome, capital R, capital R. So now what are we looking at? We are looking at generation number 2, generation genotype for generation 2 and phenotype for generation 2. And we are only solving for the first type of a genotype here, capital R, capital R. And the outcome is capital R, capital R, capital R, capital R, capital R, capital R, and capital R, and capital R. And we see here, can you see it very clearly that the recessive allelic partner is completely gone? Theoretically, we can see that, but the farmer cannot see it. The plant breeder cannot see it unless and until he self-fertilizes. So at the end of self-fertilization, what are we getting? We are getting 4 possible combinations. Now the plant breeder has to put all of these into the field. So he will again choose like 4 fields, right? He will choose, let's say, 4 fields. Let me just draw small, tiny small fields. Here is 1, 2, 3, and 4. And he'll take these particular seeds and he'll sow them here, he'll sow them here, sow them here, and sow them here. And what does he get? He gets all red plants. So what is the outcome here of the second generation? In the second generation, the outcome of one of the allelic partners is we've got 100% capital R, and in the phenotype, we got 100% red flower producing plants. But in order to confirm that, the plant breeder, what he'll do, he'll pick up one of those again, these plants. He will self these plants once again, and then he will see whether they are still producing red flowers. He will also take seeds from the other ones and self fertilize these particular plants just to observe whether they are also producing red flowers. And when the plant breeder reaches the point when all the plants upon self-pollination end up producing his choice of trait, then he can say that he has generated a pure line for that particular given trait. Well, the plant breeder could have also chosen to produce the pure line for the yellow flowers. I hope you've understood how pure lines are generated, and I hope we've succeeded to a certain degree in our discussion or in my one-way instruction. Thank you. I will see you in the next video.